Okay, so we are reviewing the scientific method. What's the first step in the scientific method? First thing you got to do whenever you're going through an experiment. What's the first thing? Remember? Observe, question, observe, or question. And once you make these observations or ask these questions, then what do? Yeah. Form a hypothesis. Correct. And a hypothesis is, remember, not an educated guess. That's an okay definition, but we're high school biology students now. So a hypothesis is a tentative explanation based on prior knowledge, previous experience, or previous research, whatever. Okay? So we make an observation, we ask a question, we come up with an answer to that question, which is the hypothesis. Then what do we have to do? Design an experiment. Design an experiment and do what while we're doing that experiment? What do we get from that experiment? Design an experiment and collect data. Okay, we've designed our experiment, we've collected our data. Now what do we do with the data? Analyze the data. Very good. And once we've looked at our data, what does that data let us do? There's a word you can use here that I don't like to use just because the scientific method is more of a cycle than a start here and end here type of thing. What do you do once you've analyzed the data? What does that give us? An answer to our question, an answer to our hypothesis. So when we get to this point, we can either accept or reject our hypothesis. So in our experiment, if we were right, we can say, yeah, we were right, but that might lead to more questions, or it might lead to more observations that lead to questions, and might have answers to that as well. The other thing that you could use here, the other word is what? Say it again? Or did somebody not say it? What word don't I like to use here? Because it seems fine. Starts with C. It's the end of something. Conclusion. Every once in a while, you will do an experiment that does have a pretty concrete conclusion that doesn't necessarily lead to another observation or question. However, most times you just keep going through this and we keep learning and developing and what have you. On the test, you'll see words like conclusion. You'll see words like prediction. Okay, which hypothesis is like a prediction. You'll see words like problem. When you see the word problem, that's like at the very beginning. New to this class, or you are? Rylan, you're not on my roster anymore, buddy. You are not on my roster anymore. Rylan Smithley, not on my roster. Did you get your schedule changed? So that's a scientific method. You'll see these other words in tests and prompts and things like that. Sometimes you start with a problem, like my fish are dying. That's a problem. Or the, sun never comes out. That's a problem, right? I don't know if you could solve why the sun never comes out, but you get the idea. Okay. Girls, can you read this? Because it's flipped on my end. Can you read it pretty
I
clearly?
can read it.
You
Yeah,
can
we can
read
read it.
it? Good. Thank you. All right. That's the scientific method. Now, we also talked about the difference between an observation and an inference. Observation, we use our 5 senses to describe. Inference is based on an observation. And an inference is really like a simple conclusion based on an observation and previous knowledge, okay? Based on observation and prior knowledge. Okay. Now, what's a controlled experiment? It's just an experiment that has only one variable. What's a variable?
A variable is the changing factors in an experiment.
Yes, it is. A variable is a changing factor in an experiment. A smaller word within variable would be vary. What's vary mean? Or, sorry, not vary, as in that modifier. Vary, V-A-R-Y. Vary means to change. Yeah, the weather varies. The weather will change. Variable is a changing factor in an experiment. Okay? There are several types of variables and other contents in an experiment. And I'm going to go through them really quickly, but I'm not going to dwell on them because we already went through them before. Then we're going to practice with them. Okay? What are the 2 main types of variables in an experiment?
Independent and dependent.
Independent variable and dependent variable. Okay? The independent variable is what we are deliberately changing. What we are changing. And the dependent variable is what we are what? Yep. Measuring. Okay? Dry mix. The reason we need dry mix is for graphing. Okay? Dry mix is when we're graphing results. Okay? D stands for dependent variable. It responds to the independent variable, and it's graphed on the y-axis. I stands for independent variable. It's graphed on the x-axis. And the independent variable manipulates, which means changes, the dependent variable. If this doesn't make sense to you, then it's not going to be helpful. Okay? The main thing it helps you with is where the dependent variable is graphed and where the independent variable is graphed. The other two words are helpful in helping you determine what the dependent variable does and what the independent variable does, but I don't think for our purposes it's as important for you to memorize. Okay? Mr. Messer noticed that the plants that are near the front of the room are dying, and the plants near the back of the room are growing well. He thinks this is due to the type of light that they are getting. In order to determine why the plants are growing at different rates, Mr. Messer decides to conduct an experiment. He will place one plant under fluorescent light, one plant under the red-blue grow lights, and one plant in the greenhouse where it can receive direct sunlight. He made sure that the plants all had the same type of soil and received the same amount of water daily. He measured their growth over 5 days. He determined that the plant under the red-blue grow light exhibited the most growth. Here is the data. Over the 5 days, the plant under the fluorescent light grew about 1/4 of an inch a day, pretty consistent. The plant that was under the red-blue grow lights, what's up? They just now put it back in here? The plant that was under the red-blue grow light grew 1 inch the first day, grew 1 inch the second day, grew 2 inches the third day, grew 2 inches the fourth day, and grew 3 inches the fifth day. And the plant that was under the sunlight in the greenhouse grew 1/2 inch a couple of days, 3/4 of an inch the next couple of days, and 1 inch the next day. Now, let me ask you something just about reading and interpreting the information here. How many inches did the plant grow that was under the fluorescent light? How many inches total? After 5 days, the plant that was under the fluorescent light grew how many inches? 1 1/4 inches. It grew 1/4, 1/2, 3/4, a full inch, and 1/4. All of those added together. It grew that much each day. How much did the plant grow underneath the red-blue grow light? 9 inches. So the plant under the fluorescent grow light, after basically a week, only grew 1 1/4 inches. The plant under the red-blue grow light grew 9 inches. And the plant in the sunlight grew 1, 2, 3.5 inches. Okay? So looking at the data, you can't really see that, can you? It's not really super apparent. So what do we got to do so that we can easily interpret the data? What? Graph it. Absolutely. What type of graph might we use for this? We'll get to that. Let's do this first. Okay? Let's do this first. What's the independent variable? What? The type of light. Why do you say the type of light? Because that's what we are changing. The independent variable is what we're deliberately changing. You said type of light because that is what we are deliberately changing. We're changing it from fluorescent, red, blue, and sunlight. Very good. What's the dependent variable in this example? How do we determine the dependent variable? What question can we ask ourselves? What are we measuring? And what are we measuring in this example? Yeah. The amount of growth. Yeah. The height. I understand what you mean, but let's phrase it because it may, droop down. That's how we're going. So the dependent variable is what we're measuring, and we're measuring the amount of growth. In what measurement? In inches. Okay. Measuring the amount of growth in inches. What was the hypothesis? What was the hypothesis? Say a little louder. The hypothesis and the question are a little bit different. The question or the problem, so let's do this. The problem / question is, why are some plants dying? But what's the hypothesis? What is the tentative explanation for what's going on? What word can we look for that helps us determine what a hypothesis is in an experiment? I bet if I read the first 3 sentences, you can tell me what the hypothesis is. Mr. Messer noticed that the plants near the front of the room are dying and the plants near the back of the room are growing well. He thinks it's due to the type of light they're getting. What's the hypothesis? Correct. The hypothesis is the type of light is affecting plant growth. Right? He thinks the type of life or the type of light is affecting the plant growth. What are some of the constants? Yep, the soil is the same in each experiment. The water is the same in each experiment. The amount of water. What's the control group? This one's a little tricky. Remember what question we can ask ourselves to determine what the control group is. What's that question, first of all? Which what? Typical. Very good. What typically happens? What's normal? Okay. In this experiment, what the control group would be. Go ahead. You're correct. Why? Very good. Emily, right? Emily said the control group is what typically happens. In our experiment, the plant that grows in the greenhouse is getting sunlight, which is what typically happens. What plants normally get is sunlight and water from rain, okay? So our control group is the plant and sunlight. Okay? We've identified what the problem and the question is, what the dependent and independent variables are, what the hypothesis is, constant, control group. What about the conclusion? What's the conclusion? Do we remember? Yeah. He measured their growth for 5 days. He determined that the plant under the red-blue grow light exhibited the most growth. Most growth. So the conclusion is red, blue, light promotes the most growth. Okay. Now that we've put all that into practice, What type of graph will we want to use in this example? Will we want to use a pie chart? Will we want to graph the data using a pie chart? No, why not? We're not dealing with percentages, okay? Could we use a bar chart? We could use a bar chart, but there'd be a lot of bars, and we would preferably only graph how much they actually grew. We'll do two different charts. We'll do a bar chart second. We'll do a line graph first. We'll do the line graph first because it doesn't necessarily always follow the dry mix information, okay? So I'm going to be flipping back and forth between the data and our chart here. When you're doing a chart, what's the first thing you need to do? Title. So this is types of light and plant growth. And dry mix is dependent on the y-axis, independent on the x-axis. So dependent on the y-axis, what we're measuring, and independent, what we're changing on the y-axis. In a line graph, time is typically on the x-axis, okay? Time is typically on the x-axis. So we'll put time down here. And what's our time frame? What's the time in our experiment? 5 days. So we'll put day 1, day 2, day 3, day 4, day 5, and we'll title this axis, we'll make this axis label number of days. Okay, and then so dry mix, what goes on the y-axis? Dependent, what are we measuring? In our experiment, what are we measuring? What are we measuring here? Plant growth in what? Inches. So we'll put plant growth in inches over here. And our minimum was like 1/4 of an inch, and our maximum was 3 inches, but really our maximum was 9 inches total, and 1 1/4, and blah, blah, blah. So we'll do 1/2 inch 1, 1/2 inch 2, 1/2 inch 3, 1/2 inch 4, 1/2 inch 5, 1/2 inch 6, 1/2 inch 7, 1/2 inch 8, 1/2 inch 9. 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay. So before we've even graphed any data, we've made a title at the top, axis labels on both sides. We've determined our scale, which is days. And we've determined our scale over here, which is growth in inches, okay? Then we just have to flip back and forth from the data and plot it, okay? The fluorescent light, the plant under the fluorescent light grew 1/4 of an inch each day. So on day 1, this is a 1/2 inch, so it grew 1/4 of an inch. Day 2, got up to a 1/2 inch. Day 3, 3/4 of an inch. Day 4, an inch. Day 5, an inch and 1/4. Okay? That's fluorescent light. The sunlight grew 1/2 inch 2 days, 3/4 of an inch the next 2 days, and an inch the third day. So the sunlight on day 1 grew 1/2 inch. On day 2 grew another 1/2 inch. On day 3 grew 3/4 of an inch. On day 4 grew 3/4 of an inch from an inch and 3/4 or an inch and 3/4 was 2.5. And then grew a whole inch from 2.5 to 3.5 on the 5th day. That's sunlight. And under the red blue light, we grew 1, 1, 2, 2, and 3. So we started at 0, went to 1. Grew another inch. Oh, that's a 1/2. Sorry. 1. Grew another inch on day 2. Grew 2 inches, day 3, I think. Yeah, 2 inches, day 4. And 3 inches on day 5. That was red blue. Okay. That's a lot, isn't it? This is one problem. We define all the variables. What's the question? Hypothesis, constants, independent variable, dependent variable. Then we got to graph it. The value of doing this graph is, well, which one exhibited the most growth? Well, biggest line, red, blue, easy. Which one exhibited the least? Fluorescent. How about halfway in the middle? Sunlight. There you go. Okay. There would be another way to do this if we did a bar graph, except we wouldn't do it over time. We would do it in total. Okay. How could we put this data in a bar graph? How could we do this in a bar graph? I'm going to let y'all do this one. How could we do this in a bar graph? Here's our data. Dry mix. How could we do this in a bar graph? Do you have your hand up? Oh. Caitlin, how can we put this in a bar graph? We're not going to graph each data point. How could we get 3 pieces of data to put on this bar graph?
I don't know.
The first thing we did when we looked at the data was total up how much each plant actually grew, right?
Yeah.
Why don't we do that?
Would you just do the same thing as like on the, I don't know how to explain it. Like, would the bottom line, would that be, yeah, that line. Would that be time or?
Wouldn't be time, dry mix. The independent variable goes on the x-axis. The independent variable is what we're changing. I'm sorry. Yeah. Independent variable is what we're changing. We're changing the light, type of light. So what were the types of lights?
Fluorescent. Is that one?
Yep.
And
Fluorescent, sunlight,
oh,
red, blue. So remember, dye mix, the independent variable goes on the x-axis. The independent variable was the type of light. We put the type of light on the x-axis, right? Now, dry mix, DRY, dependent goes on the y-axis. What's the dependent? What we're measuring. But we don't want all these different data points. So what we're going to do is do the totals. The fluorescent light plant grew 1.25 inches. These are just notes over here. Fluorescent grew 1.25 inches. The red blue grew 2, 4, 6, 9 inches. The sunlight grew 1 inch, 2 inches, 3.5 inches. So on our scale over here, we need to go up to at least 9. Let's go to 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And this is growth in inches. And this is type of light. Okay. Fluorescent light grew 1.25 inches, right? Sunlight grew 3.5 inches. And red blue light grew 9 inches. And that's how we would do a bar graph with the data. Do you see how it's two different ways to represent the data, but we still get the same idea? We're still seeing that the red-blue light caused the most growth over time. We're just graphing totals here instead of everything over time, which is what we were doing with the line graph. Okay? I am going to